So the next section on this unit is the bacteria. So we know that bacteria are their own domain. They are prokaryotes. We know all about prokaryotes from the last section and about their replication and everything like that. So let's quickly talk about bacteria. So bacteria, they all have a cell wall, which is made of peptidoglycan. It's a hard word to say, but peptidoglycan. And we have this way of characterizing them, which is known as gram-negative or gram-positive. And what that is what the structure of their cell wall. So there's this stain that we can use that we put on bacteria, and gram-negative bacteria don't really show up under the stain, whereas gram-positive bacteria stain very, very darkly. And the reason for that is gram-negative bacteria have their peptidoglycan cell wall between two plasma membranes. So there's one right underneath it and one right above it. So the stain can't get to it very well, so it doesn't stain very darkly, so it's gram-negative. And then the gram-positive bacteria do not have a second plasma membrane on the outside of their peptidoglycan cell wall, so they stain much darker. So that's one way that we differentiate between different types of bacteria, is whether or not they're gram-negative or gram-positive. Most bacteria that are harmful to humans are, in fact, gram-negative, but some gram-positive have also shown to be. Cholera, with the bacteria that causes cholera, is Vibrio cholerae, and that one is gram-negative, but tetanus is a gram-positive bacteria, so both can make humans sick. There's also another way we classify them is based on their shapes, because there's so many different kinds of bacteria but they have very diverse different shapes. So there's 3 different basic bacterial shapes. There's spirili, which are spiral or helical shaped. This picture is in your textbook. I hope you can see this. Spirili, it's on page 374. Spirili is helical shaped. Bacilli is round, and cocci, or coccus, is round or spherical. So the different shapes of them, and then how many of them there are together gives it more of a name. So if they're pairs, it would be diplo right before it. If they're in chains, it's strepto, and if they're in clusters, it's staphylo. So if you guys have ever heard of strep throat, that's from streptococcus. So that's how we name our bacteria. Next, let's talk about bacterial metabolism. So you would probably be able to guess this. They inhabit so many different kinds of environments that they are very, very diverse. We have a couple of different kinds of them that we're going to talk about. So one basic differentiation is whether or not they need oxygen. So there are certain kinds of bacteria called obligate anaerobes, which cannot grow in the presence of oxygen. So think about this. Anaerobe means no oxygen, and obligate is like you're obligated. So they're obligated to live without oxygen. They cannot grow in the presence of oxygen. There's a few serious illnesses such as botulism or tetanus, which are obligate anaerobes. And they can infect you because they live inside the oxygen-free tissues of deep inside the human body. And then we have other things called facultative anaerobes, which can grow in or without, or with or without oxygen. But most bacteria are aerobic, and they do need the presence of oxygen to carry out cellular respiration. So one kind of metabolism of bacteria is autotrophic, so meaning they're making their own food. Photoautotrophs are photosynthetic, just like plants, and they use solar energy to reduce carbon dioxide to organic compounds, just like plants do. There are 2 types of these, those that evolved first and do not give off oxygen, and those that evolved afterwards and do produce carbon dioxide. So a couple of types that don't produce, oxygen, not carbon dioxide. A couple that don't produce oxygen are things such as green sulfur bacteria and purple bacteria, and they live in very poor oxygen or oxygen-poor environments, such as the muddy bottom of a marsh, according to your textbook. Whereas on the other hand, we have cyanobacteria, which we're going to talk about even more later, and they contain chlorophyll A and produce O2, and this is just like plants. The second kind of autotrophic bacteria are chemoautotrophs. So rather than producing their own food from sunlight, they produce their own food from inorganic chemicals chemosynthesis. They oxidize inorganic compounds such as hydrogen gas, hydrogen sulfide, and ammonia, and they use this to reduce carbon dioxide to glucose. So they take the energy from these inorganic compounds, such as hydrogen gas, etc., and they take energy from those and they use it to reduce carbon dioxide to glucose, just like plants use energy from the sun to reduce carbon dioxide to glucose. Some bacteria are capable of oxidizing nitrogen or sulfur compounds. This is very important to all environments because we need to recycle all of our chemicals. And if you've ever heard the nitrogen cycle or the sulfur cycle, we rely on bacteria as a key component of these. Then we have heterotrophic bacteria, and these are, think of we're heterotrophs, they take in organic compounds. A lot of these are saprotrophs, which means that they decompose large organic molecules into smaller ones that they can then absorb. And we call these decomposers in ecosystems. And these, again, are very important in nutrient cycling and in ecosystems and everything like that. They also have these capabilities. They're heterotrophic capabilities. They're chemoheterotrophic. We use them to help us make things such as alcohols. Ladies, if you're taking off your nail polish using acetone, that comes from bacteria. We use butter, rubber, coffee, cocoa. All these things are made using the help of bacteria. Now, a lot of bacteria have what we call symbiotic relationships. Symbiotic relationships are relationships in which two different species live together in a very, very, very close association. So at least one cannot live without the other. And symbiotic, sym means the same. So same or together. Together, they're living together. There's a couple of different kinds. There's three: mutualism, commensalism, and parasitism. In mutualism, think about it. If you are mutual, you're together. In mutualism, both species benefit from the association. So a couple of examples of this are bacteria in human intestines. For one, they have a safe place to live inside of our intestines. They are getting nutrients from us, and we use them to break down things that we can't break down. For example, we can't break down lactose, but the bacteria in our stomachs can break down lactose. We can't break down cellulose, which is found in plants, but bacteria help us digest that. And they release the vitamins that we can't get out of them. There's also bacteria that live in the roots of plants that help them absorb more nutrients than they would be capable of doing. So those are two examples of mutualism. Another second kind is commensalism, which is where one species benefits and the other is entirely unaffected. So the E. coli in our intestines, they use oxygen. So E. coli is the kind of bacteria that live in our intestines that help us out, and they use all the oxygen that's there. So we're giving them oxygen, and they're taking it all in because they need it. So they're not getting a benefit. They're just using the oxygen. But the fact that they're taking up all of the oxygen benefits all of the obligate anaerobes that live in our intestines. So there's a bunch of other bacteria that cannot live in the presence of oxygen, and they can live there because the E. coli takes up all the oxygen. Another example of commensalism is barnacles that live on marine species. So think of it this way. If you have a giant, giant, giant sperm whale, I think or the gray whale, the largest living animal on Earth, they have barnacles that live on them that stick to them and float around with them. And the barnacles are benefiting greatly because they're filter feeders, so they get fed as the whales swim around. The whales are entirely unaffected by this. So the barnacles benefit and the whales are unaffected. Then there's parasitism, which I'm sure you've heard of before, which is where one species is benefited and the other one has negative effects. So parasitic bacteria, the not good kind of bacteria that make you sick, they live inside your body, they're benefiting, they're taking your nutrients, and they're making you sick. Another kind of parasitism is what's called endospores. So this is a pathogenic bacteria, and let's talk more about that. So endospores are deadly bacteria that kind of go into this state of hibernation using endospores. So if they're faced with unfavorable conditions, meaning super high temperatures or not enough nutrients or anything like that, they will form what's called an endospore. So they are this protective heavy coat, and they remove a portion of the cytoplasm and a copy of the chromosome, and they live inside of this coat. So they just keep enough of what they need. They keep very few proteins, very few enzymes, and just one copy of their chromosome, and they encase them in this really tough coat. And because of that, they can survive in very harsh environments for extended periods of time. So they're basically protecting themselves and hibernating until favorable conditions return. As soon as they absorb water, they can germinate out of their endospore coat and come back to life and transform back into a normal bacterial cell. So this is really dangerous because it's really hard to kill them because they have these endospores. For example, you guys remember anthrax? There are anthrax endospores that are 1,300 years old, and they can cause just as bad of an infection as a new one. So they're very dangerous. And a lot of times the bacteria themselves that might make us sick are not harmful, but what they do is they release toxins that are poisonous. So, for example, your textbook talks about Clostridium tetani, which can form an endospore, and when they germinate, they release a toxin that causes the disease tetanus. You guys know if you've ever gotten cut by a piece of metal or stepped on a rusty nail or something like that, you need to get a tetanus shot so that you do not get sick from this bacteria. And it's some gram-negative bacteria release what's called a lipopolysaccharide fragment when they encounter an antibiotic. So basically, they come into contact with this antibiotic and they release this toxin. And this toxin can result in a high fever and a drop in blood pressure. So it's not the bacteria themselves that are making you sick, it's the toxins that they're releasing. Then sometimes fimbriae, remember we talked about fimbriae when we talked about structure of prokaryotes, they allow a pathogen to bind to certain cells. This is what leads to the specificity that comes with bacteria. They can only infect certain host cells. For example, the Shigella dysentery, which is what causes dysentery, is only able to stick to the intestinal wall, causes dysentery, which is severe diarrhea. They also release a toxin that increases the potential to kill you. So again, the bacteria make you sick, but the toxin make you even sicker. And then we have antibiotics, however, to help us fight off bacteria. The first ever antibiotic was penicillin, and now we have dozens and hundreds of different kinds of antibiotics, but they work in two different ways. There's two kinds. There's one that inhibits protein biosynthesis in bacteria, which means that they stop the bacteria from making any proteins. So that's pretty dangerous. If they can't make proteins, they can't keep reproducing and harming you. They can't make the toxins that are harming you. And what they do is they affect bacterial ribosomes. So this drug goes in and it inhibits the bacterial ribosomes, which doesn't harm you because their ribosomes work in a different way than ours do. So they stop the bacterial ribosomes without stopping your ribosomes. Then there's a second class, which inhibits cell wall biosynthesis. So remember that bacteria have a cell wall made of peptidic glycan. And what they do is these drugs go in and they stop the formation of peptidic glycan. So your body does not make peptidic glycan, so it's not going to be harmed, but the bacteria won't be able to replicate or make new bacteria to harm you. And the overuse of antibiotics is a hot topic these days. It can be very dangerous. It can lead to bacterial resistance, which basically means that the bacteria, as they mutate, natural selection with these antibiotics is pushing them to be more fit by being able to survive the antibiotic. So the only ones that are surviving and passing on their genes are the ones that are not harmed by the antibiotic. So now we have all these offspring of bacteria that aren't going to be stopped by your antibiotic, and those are what continue to be passed on. So that can be pretty dangerous. There's this disease called MRSA, which is a kind of staph infection. And when we first discovered it, only 3% of MRSA was resistant to antibiotics, and now 90% of MRSA strains are resistant to antibiotics, so that's pretty dangerous. Lastly, your textbook talks about Cyanobacteria, which is a very specific kind of gram-negative bacteria that has some unique traits. I'm going to let you guys read that one and be responsible for that one on your own, but a couple things I do want to say about it is that they photosynthesize just like plants, and for a really long time, they were classified as eukaryotic blue-green algae because they share so many characteristics with plants in their photosynthetic capabilities. And they were the ones that, think back to the last couple chapters, before the Earth did not have oxygen, but when these guys evolved, Cyanobacteria started photosynthesizing, they started releasing oxygen into the air, and that's where the original oxygen in our atmosphere came from. They are much bigger than other bacteria. They can be 1 to 50 micrometers in length, and they have no visible means of locomotion, although sometimes they can kind of glide around. So go ahead and read on your own about the other characteristics of Cyanobacteria, and that is all you need to know about bacteria.